Good day, my name is Alexander Johannes and I'm here at the University of Pretoria. We've got the Engineering 1 building behind me. I've got an interest, maybe as you, in biochemical engineering. Today I want to show you a bit about the setup I used for my experiment, namely the metabolic flux analysis of the fungus Rhizopus Oryzae with citrate addition, so that you can get a better idea of what everything looks like. Let's now head to the labs.
Fumaric acid can be synthesized by the fungus Rhizopus oryzae. Its double bond and its two carboxylic acid groups makes this an attractive chemical for chemical synthesis.
We are now here at one of the labs. This was the lab where we mixed or prepared all our solutions. And we've got the TSB, tryptoin soil broth, that was for the growth medium, all our various micronutrients. We've got our citrate right here. Substrate used was glucose, and the neutralizing agent used was calcium carbonate.
Advantages of using calcium carbonate are no pH control system is required during fermentation, and it results in the highest fumaric acid productivity. One disadvantage of using calcium carbonate is the possible precipitation of calcium fumarate. However, the solubility of calcium fumarate is currently believed to be around 25 grams per liter. The fumaric acid concentrations for this study were more towards 10 grams per liter, so in theory, this should not disadvantage the results.
Then, after preparing the solutions, they were autoclaved in this autoclaving machine here. After autoclaving, they were mixed and then inoculated with the cells. The cells were stored in this fridge here, obviously so that they can last longer. After inoculation, the cells were incubated in this little incubation hood here. The final product looked like this. This was after the cells were grown. They were connected in here to a pump, this pump here, to allow for aerobic conditions as is necessary for fermentation. You can also see growth plates inside this container here so that the cells could attach to something and grow properly.
All fermentable carbon sources in the fungus Rhizopus oryzae are metabolized to pyruvate. This is subsequently transferred to ethanol under anaerobic conditions and to fumaric acid under aerobic conditions. The cell development is as per the following pictures. You can clearly see that the solution clears up as the fermentation proceeds. This is because the neutralizing agent gets consumed. For one citrate concentration, 2 grams per liter, a different cell formation was observed. Pellets were seen to have formed instead of the usual mycelial cell development. This was a clear indication that citrate has some sort of influence on the cell development.
I will now quickly show you the hood where we did all the transferal of the stuff under sterile conditions. This is the air hood. All transferal of mediums or cells, inoculations, everything that could expose the cells or the medium to the atmosphere was done under this hood closely to this plane to prevent any infections from entering the medium. Once we prepared the medium and fermentation proceeded, samples were taken over a period of 5 days. The samples were then analyzed with this high-performance liquid chromatograph on a daily basis. One experiment was done slightly differently. All the experiments to prepare the fermentation medium, 2 solutions were prepared initially. The one solution contained the glucose, the substrate, and the other solution contained all the micronutrients, including the citrate for the citrate experiments. Those 2 solutions were prepared, they were autoclaved separately, and then after autoclaving they were mixed and the grown cells were added to this. So for this particular experiment I decided to add the citrate to the glucose solution instead of the micronutrient solution before autoclaving.
The HPLC analysis of this differently done experiment is quite interesting. You can see it on this image. There is clearly some formation of an unknown sugar during autoclaving.
That was a quick tour through the laboratory. I hope you got some sort of indication of what the project was about and what was done, and you could follow visually the experimental procedure and all the chemicals used. Thank you for your time and enjoy the rest of your day.